And the next speaker is C4U. The speaker is Mr. Kohei Kishi. And please wait for a while. We are preparing the slides. Okay, please begin. Thank you.
Okay, so thank you very much. So I'm very glad to be here to introduce my company, C4U, as well as about technology, CRISPR-Cas3. So some of you are familiar with CRISPR gene editing technology, and that is a CRISPR-Cas9, but our technology is CRISPR-Cas3. So I will explain what is the difference between the Cas3 and Cas9 today. So C4U stands for the CRISPR for you. So I think that you don't remember CRISPR, but please remember C4U. So our company was established in 2018, 6 years ago, from Osaka University. So Professor Mashimo, who was in Osaka University, he discovered CRISPR-Cas3, and he established the company C4U. And now he moved to the University of Tokyo, so that we have the 2 research laboratories in Osaka University as well as the University of Tokyo. And CEO and President CEO is Aki Hirai. And he joined the company 4 years ago, and he is not the founder, but he is an expert of the managing the company. And our CFO is Sonoda. He is a CPA holder, and he is also the board member. And DCIP head and Narita-san, he is also the board member of the C4U. So this slide is very important for C4U. What is the difference between the CRISPR-Cas9 and Cas3? So we have identified at least 3 big differences between them. Number 1, the high selectivity. So as you may know, CRISPR-Cas9 utilizes 20 bases to recognize where to delete in a gene. So that means 4 to the 20th powered selectivity. On the other hand, our CRISPR-Cas3 utilizes 27 bases. So 4 to the 7th power more selectivity by using the CRISPR-Cas3. At this moment, we do not have the off-target event. Off-target means the undesired deletion of the gene. The second point is the type of the deletion. So CRISPR-Cas9 can cut the gene like a scissors as a point cut. On the other hand, our CRISPR-Cas3 can delete a larger portion, like a shredder. So that is a quite big differences. So if you consider the completely knockout of the gene, our CRISPR-Cas3 would be more appropriate than CRISPR-Cas9. And last but not least, the IP is quite different. That is a different point. So as you may know, CRISPR-Cas9 discovered 2012. At that time, a lot of organizations insisted the ownership of the CRISPR-Cas9. For example, Broad Institute and UC Berkeley, and also the Vilnius University, they are insisted that is ours. On the other hand, our CRISPR-Cas3, as I mentioned, Osaka University discovered this technology, and we received exclusive license from Osaka University. So if you consider the business by using the CRISPR-Cas3, please contact me. And importantly, there is no overlap pattern between the CRISPR-Cas3 and Cas9. It's very completely separated, so it's very good. So considering such kind of profile for the large deletion, these indications will be the appropriate indications. And mucolipidosis, actually we are developing CRISPR-Cas3 for this indication, and also the CNS repeat disease, and beta thalassemia or the SCD, and also the DMD, also the target indications by using a CRISPR-Cas3. So these are pipeline. So as I mentioned, we have the 2 business models. One is our in-house pipeline. So we are developing the mucolipidosis with the National Center on Child Health and Development in Japan. And currently, we are in the discovery stage, and we are planning to the human in 2026. And also, our research team is ongoing to develop the second asset At this moment, we cannot disclose it, but we are seriously discussing our researching activities. And also, we have the several partnerships with the venture company and also the pharmaceutical companies. For example, we have the research collaboration and license agreement with Noiroimmune. They are the CAR T company. So they utilize our CRISPR-Cas3 for T cell. So gene-edited T cell is modified or utilized for their CAR T platform. So that's one example. And we have the several collaboration with Sumitomo and top Japanese pharmaceutical company. So in the Sumitomo case, we had a research collaboration agreement with them, and this April, they got a good result during the research collaboration, and they have exercised the option to enter the next stage as a licensing agreement. And not only the Japanese company, but also we have the Switzerland Hilaiba company, that is a venture company for the dermatology area. So both of us are developing epidermolysis bullosa for the very rare diseases. And also we have the several collaboration with Sereido or CDMO SiteFact. And yes, so we have received Series B financing. So last May, 11 million US dollar, 15 億円, 15 Japanese 億円. But we are planning to the next Series C early next year or mid next year. So not only the pharmaceutical area, but also we have the several collaboration with other industries, such as plant, agriculture, or fish, or microalgae area. So as you may know, so genes are located in any creatures. So not only the human, but also the plant or animal or fish. So thank you very much for your attention.
All right, thank you very much, Mr. Kishi. Okay, Q&A again, if you have any. Okay, gentleman from the second row from the last. Okay.
Thank you for your excellent presentation. I have a question about your in-house pipeline. So why did you choose to target mucolipidosis? And are there any competitors developing similar drugs?
Thank you very much. So as you may be familiar with that disease, so it's a very rare disease. So at first we would like to establish or show our efficacy in a human as soon as possible. So we have selected such kind of diseases, the short period. So at this moment, it's very rare and the number of the patients is very small. So we will try to expand our technology to the next disease. So at this moment, I do not understand the direct competitor because it's very, very rare disease. Is that okay for your question?
Okay, thank you.
You are welcome.
And one more question from the front. Any other question? Okay, please.
Yeah, Christopher Cas3 technology is very fantastic because no overlap the patent, and but around these technologies, you need to some drug delivery systems. And how about the drug delivery systems patent? How do you handle such problems?
Thank you very much for your question. So at this moment, so for example, mucolipidosis. So we are developing it, so CRISPR-Cas3 for the mucolipidosis by ex vivo setting. So hematopoietic stem cell is extracted from the patient, and outside the body, we conducted a gene editing platform and return to the patient. So that way the extraporation system was conducted. But near future, we need to consider in vivo setting. So unfortunately, this CRISPR-Cas3 is larger than the CRISPR-Cas9. So So as you may know, CRISPR-Cas9 utilizes adeno-associated virus AAV to carry the CRISPR-Cas9. So that white virus is not available for the CRISPR-Cas3. So we need to consider the other virus or non-viral delivery systems such as LNP, ribonucleoprotein particle. Yes. So so currently, so BD team, including me, is contacting the several companies for the such kind of delivery system. So C4U doesn't have such kind of delivery system by ourselves. Thank you. You're welcome.
Right, thank you very much. We may move on to the next presentation. So thank you very much, Mr. Kishi, again.
Thank you very much. Thank you very much.